Alright, good morning everyone. You're welcome to today's class. And we will continue from where we stopped in our last class. Today, under our work of genetics and hereditary, we'll be looking at DNA replication and genetic coding very briefly before we continue. Now, it is important to note that the basis for transmission of characters is the DNA, and the DNA is located where? The chromosomes, and chromosomes tend to aggregate as strands. You find chromosomes as strands, and along these strands there are various points of the chromosome that are called loci, and the loci is where a particular gene for a chromosome is found. Like in the human system, we have about 50,000 genes. And some of them are sorted independently while others are sorted separately. Like we have 23 pair of chromosomes of genes that are sorted together. And that tells us that chromosome is actually one of the bases for inheritance. And within the chromosomes, we have this DNA, and the meaning of DNA is what they also ribonucleic acid. And it tells us that genes are transmitted as chemical substances, as chemical compounds, precisely as amino acids. So it is these amino acids that codes for various traits and characters. An amino acid that is synthesized is what tells the difference between the manifestation of one character and another. For instance, in sickle cell anemia, we have one of the amino acids is replaced with another. We have one of the amino acids that is found in the red blood cell being replaced in miscoding due to mutation. That miscoding is what leads to the formation of that sickle cell. So therefore it tells us that amino acids are important in transmission of genetic character. And we say that the DNA strand is held by what? Weak hydrogen bond. So the kind of bond you find between DNA strands. We don't have a strand of DNA, assuming we have a portion of DNA here. The strands are actually found as anti-parallel. The DNA strand is actually anti-parallel in nature. Okay? Sometimes you can draw it as something of this nature. Anti-parallel. The strands, they are not completely of this shape, they are anti-parallel. And inside, in between that DNA strand, we have various amino acids of this nature, and they are held in position. They are held in position by a weak hydrogen bond, and that tells us that this hydrogen bond can easily be unzipped during formation of gametes. So we said DNA strands is held by weak hydrogen bond. They tend to unzip accompanied by replication of nucleotides during meiosis. Meiosis, which is what? Reduction division. Reduction division. There is tendency of the nucleotide that is formed to unzip. When it unzips, when the DNA unzips, the nucleotides recombine themselves and it leads to the formation of two DNA strands. That is how DNA replication happens in any living tissue. And we say that hydrogen bond is formed again back when these complementary strands. There are strands that are called complementary strands, meaning when you find one strand, the other strand is bound to combine with it. And that's why we call them complementary strands. They go hand in hand. Okay? The next thing we look at is the genetic code. For every individual, for every living organism, there is an array of genetic coding that is particular to a particular class of organism. And this information carry how the information of how the organs in that person will turn out to be, like the body color, like facial and the shape of the face, and all those tiny information are found in what the genetic code is an information. So what makes a human being different from, let's say, a dog is the genetic code of that human. So when the humans start developing, because of the synthesis of varying amino acid, they will develop gradually to become completely different. So all the information that is needed for in an individual is found in its genetic code. And earlier, as at 2003, so around that time, there was an experiment. There was an experiment that was carried out to determine the genetic mapping of humans, and it was done to find the various locations, the various loci where you can find certain traits and characters. So if we look here, you find out that adenine, anywhere you find adenine combines with what? Thymine. Okay? So we call these what? Complementary strands. So what's the complementary strand for adenine is what? Thymine, and it's thymine, and that of cytosine is what? Guanine. So they combine together in a particular DNA strand. Now, the next thing I want us to take note of is what is called triplet codon. Now, amino acids combine. If you observe, you find out that adenine, thymine, cytosine, and guanine, they are all amino acids. Do you understand? These are amino acids, but these amino acids tend to combine in forms of triplets to synthesize functional amino acids in the body. Like when we have here, we have adenine, and what? Thymine. What you have formed is what? Leucine. Where you have cytosine, guanine, and adenine, you have what? Alanine formed. And when you have what? Cytosine, and adenine, you have what? Glycine is formed. Do you understand the simple basis of genetic combination? So these is what we call triplets, and these triplets also have their complementary aggregation. They complement each other. So you find that when you have leucine synthesized, alanine is also synthesized, and glycine is also synthesized. So this is just like a brief summary of DNA replication and genetic coding. In continuation, I want us to talk about the subject of amendment of Mendelian genetic theory. Now, Mendel proposed two laws of genetics. One is what? The law of what? Segregation of gene, and the law of what? Independent assortment of gene. The law of segregation of gene and independent assortment of gene. Now, by modern discovery, first of all, according to Mendel also proposed the chromosome theory of genetics, and it helped to prove, when we did the topic of evolution, it helps to prove Darwin's theory of evolution. But there was another scientist by name Thomas Morgan that helped to illustrate. What he did was he helped illustrate the fact that chromosomes are the basics for genetic variation. Now, there are certain amendments that were made to Mendelian genetic theory, Mendelian genetics. And the first one we'll look at today is incomplete dominance. The subject of incomplete dominance. According to Mendel said that a gene is either what? Dominant, or it's what? Recessive. A gene is either dominant or recessive. But when we take a pure stock of red flour, 4 o'clock flour, if you have this 4 o'clock flour, and 4 o'clock flour actually produce two types of flour. Some of them are white stock of flour, and others are red. That's why it was suitable for this experiment. When a pure stock is a what? Homozygous red. Do you understand? Like we have capital R, that's a homozygous red. When a homozygous red flour was crossed with a homozygous white flour, the resultant, actually, by normal crossing, what we're supposed to have is what? Or cross a homozygous red. And since the homozygous red is dominant, capital R, or cross a homozygous red with a homozygous white, we're supposed to have something of this nature, right? RR, and RR. Meaning we have what? 4 ratio. We have 4 heterozygous what? 4 heterozygous what? Red. They will be red in color. Okay? And this is for why we can say that for this particular flower formation, we can say that the phenotype, we say that the phenotype is what, the genotype rather, the genotype is what we see. We can say that the genotype is complete pure breed of what? Heterozygous red. Okay? But when this crossing was done, we had this is what we have formed by genetic crossing. But this color that was formed, we said this is what? Homozygous what? Red and what? Homozygous white. When this crossing was made, it was found that the crossing produced 4 homozygous color, but this time the color that was formed was not red, neither was it white, it was pink. And pink is an intermediate between what? Red and white. So we said this was incomplete what? Dominance, meaning the white is still there, the red is still present in the same flower. So both of them are sharing dominance, and that's why you have the what? A pink color, an intermediate between red and white. So scientists we went further to carry out the next generation process, which is the second filial generation. And if you carry out the F2, you find out the F2 generation by crossing two heterozygous, which is RR and RR. What are we going to have? We're going to have this one. Okay. So what you have here, the color that is formed, this will be what? Red, and this two will be what? Pink, and this will be what? White. And in the ratio of what? 1:2:1. This is the observed genotype, and it's no different from the phenotype that will be observed. The phenotype that will be observed will also be what? 1:2:1. Do you understand? Phenotype is, we said the phenotype is what is seen, the physical manifestation, what's the appearance of that particular gene combination. So the phenotype and the genotype will be what? 1:2:1 for the F2 generation. Unlike the Mendelian crossing, which will tell you that the phenotype that will be formed will be what? 3:1 according to Mendel. So this was a deviation from the Mendelian genetic theory, and that's one of the amendments, incomplete dominance. The next thing we're going to be looking at is the idea of co-dominance.